Medicine Rest Briefing. My name is Sylvia Adams. I am an assistant professor of medicine at the New York University School of Medicine and specialize in the immunotherapy of cancers and the care of patients with breast cancer. As a reminder, all research presented today is embargoed until 12:01 a.m. Monday, June 4th. Today's featured studies show the promise of personalized medicine. Personalized medicine, or precision medicine, is a patient-focused approach that targets treatments to the genetic abnormality in cancers that drive cancer growth and the metastatic behavior. The successes that you will see today resulted from translation of scientific discoveries to very sophisticated clinical trials, and in our case here, being performed in over 1,000 patients worldwide. The first 4 studies promise to expand the range of effective and targeted therapies for patients with common cancers such as non-small cell lung cancer and melanoma, but also for patients with rare tumors such as the gastrointestinal stromal cancers. The last presentation will then nicely sum it up and affirm that patients in all settings can be tested for their genetic abnormalities and cancers and can expect to benefit in the coming years. So our first presentation will highlight the results of an international phase 3 study evaluating an oral targeted drug, Regorafenib, in patients with gastrointestinal stromal tumors that progress on current FDA-approved targeted treatments, imatinib and sunitinib. And I would like to introduce Dr. George Dimitri. He is the director of the Ludwig Center at the Dana-Farber Harvard Cancer Institute and senior vice president for experimental therapeutics at the Dana-Farber Cancer Center in Boston.
Thank you. Thank you all for coming. And in my 5 minutes speed dating with you, I'd like to share with you some of the exciting data that my colleagues and I worldwide have been able to generate for this unmet medical need. As many of you know, over the last decade, we've developed GIST from an unknown disease to a very widely known, more adequately treated disease, a form of sarcoma where the basic scientific understanding has truly translated into revolutionary improvements in patient care. We only have 2 FDA and worldwide approved drugs, imatinib, known as Gleevec, and sunitinib, known as Sutent. These are the only 2 drugs to treat this. These are oral pills which inhibit the mutated, activated cause of the disease. 2 different enzymes, one called KIT in a smaller group, an enzyme called PDGFRA. These drive GIST, and by inhibiting the abnormal signal, we've increased patient survival with patients with metastatic disease from before the year 2000, 3 to 6 months, to where we are now, 5 years or more. I'm still taking care of patients who have been on imatinib or Gleevec for 12 years. Only about 1 in 10, however, is that lucky. For 9 out of 10, resistance occurs, and with the emergence of drug resistance comes a fatal outcome eventually. So some years ago, we were studying various small molecules to try to find one that might shut down the target to which this tumor is addicted, this enzyme called KIT. The tumors are truly addicted. You can't see this very well, but this is another small molecule that inhibits the target kinase KIT to high potency and inhibits a number of other things like the angiogenic VEGF receptor and a few other molecules there. We've done some predictive preclinical testing, showed it looked interesting, and in a unique academic industrial collaboration, got the drug from the people who made it and discovered it, Bayer Pharmaceuticals in Germany. And my academic collaborator and I wrote an academic multi-center phase 2 study that began in February 2010 and presented positive data at last year's ASCO. On the basis of that, we were writing the phase 3 study. And here is the phase 3 study that Bayer allowed us to write and then execute as a collaborative study, multi-center, randomized, placebo-controlled, worldwide, 17 countries across Europe, North America, Asia Pacific, where patients were stratified by whether they only had the 2 approved therapies, Gleevec and Sutent, or whether they had Gleevec, Sutent, and then a whole bunch of other things afterwards. And then we also stratified by whether it was just Asia or the rest of the world. Patients with metastatic or unresectable GIST after failure of at least imatinib and Sutent were randomized in a 2-to-1 way to get this drug, Regorafenib, or a matching blinded placebo. Everybody got best supportive care. The schedule, 3 weeks on the drug, 1 week off, repetitive cycles. And then we followed them. We watched them very carefully, watched their x-rays, this blinded central view, tried to take out any bias from the system. And if a patient progressed, we offered that patient unblinding, and if they were on the placebo, they got to cross over to unblinded active drug, Regorafenib. If they were on Regorafenib, but the doctor felt it was still benefiting them, those patients could also continue on the drug. So at that point, patients were on unblinded Regorafenib. About 85% of our placebo patients ultimately crossed over. At the next progression, however, patients came off treatment and off study. Here are the data. The progression-free survival difference was highly statistically significant. The Regorafenib induced almost 5 months of disease control compared to less than 1 month time to progression in the group that got the placebo. This is a hazard ratio of 0.27, meaning a 73% decrease in the risk of progression or death with the drug compared to placebo, and basically a positive study on the basis of this. Now, the disease control and overall response rate, this essentially measures something like that. The durable, stable disease rate of people who had control for more than 3 months was more than 50% in the group that got the Regorafenib and less than 10% in the placebo-treated group. Note here that the objective shrinkage of tumors was small. And we expect that with many of these drugs for a variety of biological reasons. We understand the targets. We're understanding better how we can control the disease and give patients benefit, even if we're not shrinking the disease. Even in the active drug, less than 5% of patients shrank their tumors by standard metrics of so-called response. Survival would be confounded. As I told you, 85% of patients crossed over to the active drug, so if the drug works, it will make them live longer, and that's exactly what we saw here. The hazard ratio for overall survival trended in favor of the patients who got started on the Regorafenib, a 23% decrease in the risk of death, but that was not statistically significant, as expected, because patients got to cross over and essentially have their lives presumably prolonged by getting the active drug. So that was part of the study design. In terms of side effects, these were as expected. There were no significant grade 4, grade 5 differences between the placebo and Regorafenib. However, if one looks at the all grades, there are many more minor grade 1 and grade 2 toxicities with the Regorafenib, but a not insignificant number from, let's say, 10 to 27% in the placebo group, showing just how symptomatic many of these patients can be with advanced treatment-resistant GIST. The 3 most important grade 3 or more serious toxicities are hand-foot skin reaction, a rash and potential blisters on the hands and feet, high blood pressure, hypertension, a class effect with VEGF receptor drugs, and diarrhea, which we've expected, all manageable with the appropriate dose modifications built into the study. Now, here's the precision part of this. We've understood that GIST is molecularly different diseases, and we've got other exploratory endpoints in this looking at the specific point in the GIST gene that's mutated. The most common place it's mutated in KIT is exon 11, and if we look at that, the hazard ratio for that group is 0.21, so a significant benefit in favor of the drug compared to placebo. The second most common group is exon 9, a very different mutation in the kinase. I don't have time to get into the biochemistry, but importantly, we see essentially the same benefit in those patients with a hazard ratio of 0.24, let's say, 0.239. So the benefit is preserved across these subtypes. So in conclusion, we see this as effective on the basis of this large phase 3 prospective study, which accrued very rapidly. 7 months to finish a global study in a rare disease is a wonderful thing for our global community. This has unique activity to benefit patients after resistance to the only 2 drugs available by prolonging disease control. The crossover trial design allowed patients to participate while minimizing the risks of being assigned to placebo, and I think that's important for patients to understand. We care deeply about the participants in our studies. This is a very nice study design, and this represents a unique academic-industrial collaboration with a global collaboration of investigators with very fast timelines. I'll point out that the very first GIST patient in the world to get this drug on our academic study was in February 2010. The phase 3 study rapidly accrued worldwide, and we have positive phase 3 trial results that will go to regulatory agencies by March 2012. Very rapid drug development because of a unique academic industrial collaboration. We don't get any money from it, mind you. There's no stock, there's no royalties, none of that. I just mean that there was a real mutual respect between our industrial colleagues, 2 of whom are sitting in the room from Bayer, and the academic community worldwide. So with that, thank you for your attention.